Hello students, Manjunath here. So let us start with the next part of the chapter in this particular video. Okay, the name of the chapter is the principles of inheritance and variations. So coming to the next part of the chapter, that is pedigree analysis. So what exactly is meant by pedigree analysis, right? Pedigree analysis is the study of ancestral history of a man, of transmission of genetic characters from one generation to the next generation. Okay, it is also what referred to as analysis of traits. Okay, pedigree chart or pedigree analysis can also be referred to as what? Analysis of traits in several generations of a family. Okay, analysis of traits in several generations of a given family is known as what? Pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over different generations. Okay, in the pedigree analysis, what is done? Analysis of inheritance of a particular trait is represented. Okay, and that particular trait is represented which to of a particular family, and that is why it is called as family tree. Inheritance pattern of a particular trait as a family tree over different generations, right? That itself is called as pedigree analysis. And in the pedigree analysis, the family tree will be represented using some of the symbols, and those symbols are nothing but what? Okay, these which are given in this particular photograph, right? The first one, male. Okay, normal male will be represented by a square and a female will be represented by a circle. Mating between a male and female will be represented by square and a circle which are joined by a line, and their children will be represented by the particular characters such as the tree, okay, by means of representing a tree, okay, or else the branches of a tree we can represent the children. And dizygotic twins can be represented by this particular symbol, right? Next one, monozygotic twins will be represented by showing the attachment line between those two, right? Sex unspecified will be represented by a rhombus, right? And here if you write any number in these particular square or the circle, that represents the number of children of that specific sex. Here 2 in the square represents 2 male individuals, 3 in the circle represents 3 female individuals, and if they are darkened then it is called as affected individuals. Okay, a square or a circle, if it is darkened then it is affected. If it is partly darkened like this, it is called as heterozygous and autosomal recessive. Okay, heterozygous for what? An autosomal recessive trait. Then if a dot is kept inside, then it is carrier for a particular recessive trait, right? Next one, death of the individuals can be written like this. Next one, abortion or stillbirths are written like this, which will be darkened inside, right? And the propositus will be represented by giving an arrow mark there. Next, the method of identifying persons in pedigree. Here the propositus is child 2 in the generation. See the propositus, it will be shown by the arrow mark like this. Then consanguine marriages. Consanguine marriages are represented by drawing 2 lines between the individuals. Okay, consanguine marriage. So what is meant by consanguine marriage? It is marriage within the family. Okay, marriage within the family members, that is called as consanguine marriages, right? And let us go to the next one, the rules for pedigree chart analysis. Okay, rules for pedigree chart analysis. So these are the rules for study of a pedigree chart. Okay, if you want to study any of the pedigree and if you want to know the trait, okay, whether it is autosomal recessive or whether it is autosomal dominant or X-linked recessive and X-linked dominant, or whether it is Y-linked. 5 different types of what the inheritance patterns are shown by the characters, and to know the inheritance patterns you have to know some rules about it. Okay, if any of the character is given, it will be of any of these 5, and if you want to study any of the character, or if you want to identify whether it is sex linked or autosomal linked, dominant or recessive, or it is Y linked, you have to know these particular rules, right? And in the autosomal recessive trait, see what are the rules? It appears in both the sexes with equal frequency. If the trait is appearing in both the sexes, that is male and female, with equal frequency, then it may be autosomal recessive trait. And it also have the following characters. See, the traits tend to skip the generations. Skipping of the generation is seen in case of recessive trait. Do remember, very important point it is, in case of recessive traits, skipping of the generations are seen. Next, affected offsprings are usually born to unaffected parents. In case of recessive trait, very importantly, the affected individuals will be generally born to what? Unaffected parents. And here unaffected parents means they will be carrier. Okay, they will be carrier. It means they will be heterozygous for that particular trait. Next one, when both the parents are heterozygous, approximately 1/4 of the offsprings will be affected. If both the individuals or both the parents are heterozygous in nature, approximately 1/4 of the individuals will get the affected, right? Next one, it appears more frequently among the children of consanguine marriages. Okay, the autosomal recessive traits will appear more commonly in the children of what? Consanguine marriages. That means if the individuals are married within the family, okay, if the individuals get married within the family members, then the frequency of getting the autosomal recessive trait will be more. Next one, autosomal dominant trait. It appears in both sexes with equal frequency again, but both sexes transmit the trait to their offsprings. Okay, both sexes transmit the trait to their offsprings, and it do not skip the generation. Very important point here. In case of dominant trait, skipping of generation is not seen. Okay, they will not skip the generation. Next point, affected offsprings must have one affected parents at least. Okay, if any individual is affected by any of the genetic character, then at least one of their parent should be affected, unless a new mutation is occurring. Okay, unless they possess a new mutation, they should have one affected parent. Then fifth one parent is affected and other parent is unaffected. Okay, when one parent is affected and other parent is unaffected, then what will happen? Approximately half of the offsprings will be affected. In case of dominant condition, if one parent is affected and other parent is unaffected, approximately half of their children will be affected. And the last one, unaffected parents do not transmit the traits. Very important point here, unaffected parents. That means here there is no possibility of any carrier. Okay, that is why unaffected means they will not be any carrier and they will be completely normal. They cannot transmit the trait to any of their offsprings. Right. Next one, sex linked recessive. In that particular X linked recessive trait, here more males than females are affected. Very important point, because in case of females, the X chromosome will be having another homologue, but in case of males, the homologue will not be there, they will have Y chromosome. That is why more males than females are affected. Next one, affected sons are usually born to affected mothers, because mothers will transmit X chromosome to the son but not the father. The father will be giving X chromosome to only of their daughters. If they give the Y chromosome, then it will become a male individual. That is why affected sons are usually born to affected mothers. Right? Hence, the trait can skip generation. Right? Next one, a carrier or heterozygous mother produces approximately half of the affected sons. Okay, carrier mother produces approximately half of the affected sons. Next important point, it is never passed from father to the son, because father cannot give X chromosome to the son. He will give Y chromosome. Next point, all daughters of affected fathers are carriers, because father will always give what? X chromosome to the individuals. That is why the daughters will get carriers. Is that clear? And X-linked dominant trait. Here both males and females are affected. Often more females than males are affected. X-linked dominant, because the possibility of having X chromosome in the females is more when compared with the males, because male will have only one X chromosome, but in case of females 2 X chromosomes will be there. That is why the possibility of getting X-linked infection or X-linked affected condition in the females will be more. And it does not skip generation again. Okay, it does not skip the generation, because in case of heterozygous condition also it will show the affected condition, right? That is why does not skip the generation. Next point, affected sons must have an affected mother, and affected daughters must have either affected mother or affected father, because both mother and father can give rise to what? The X chromosome to in their daughter. Next point, affected father will pass the trait on to all their daughters, because affected father will always give what? X chromosomes to their daughter. Okay, that is why they will transfer the trait to all of their daughters. Since it is dominant, all daughters will get affected. Next point, affected mothers, if heterozygous, will pass the trait on half of their sons and half of their daughters. Okay. Next one, Y-linked inheritance. It is found only in the male because Y chromosome is present only in the males. Right? It is passed from father to sons and does not skip generation here. Okay. It will be passed from father to the son and there will be no skipping of generation. So these are the rules that you have to remember when you start solving any of the pedigree chart or when you start analysis of any of the pedigree chart. So let us go to the next one, that is gene disorder. Okay. Gene disorder or the genetic disorders. Okay. Genetic disorders. What do you mean by a genetic disorder? Genetic disorders are those disorders which are related to the gene or genetic content of an organism. Okay, genetic disorders are the disorders where they will be affected with or they will be showing any defect in their genetic material. That is why these disorders are called as genetic disorders. So next one, see here, a genetic disorder is a disease that is caused by abnormality or the fault or defect in what? Individual's DNA or genetic material. And these genetic disorders are basically categorized into two categories or two groups. That is, first one is Mendelian disorder or Mendelian or gene disorders. They can be called as Mendelian disorders or they are also called as gene disorders. Okay, Mendelian disorders or else gene disorders, because Mendelian disorders, since they are called as gene disorders, because the defect will be present in only one gene, only one particular gene. The defect or the mutation or else any problem or abnormality we seen, okay, abnormality is seen with respect to only the particular gene, only one particular gene. So that is called as gene disorder, right? And next one, the chromosomal disorder. Chromosomal disorder means the disorder will be or the abnormality will be seen in the complete chromosome. Okay, if the abnormality is seen in a complete chromosome, it is called chromosomal disorder, and if an abnormality is seen in the gene, then they are called as gene disorders or Mendelian disorders. So let us go through these one by one. Right, coming to the first one, Mendelian disorders. Okay, Mendelian disorders. So as I have already told you, Mendelian disorders are mainly determined by alteration or mutation in single gene. That is why they are also called as what? Gene disorders. Do you remember, Mendelian disorders are the disorders which are mainly determined by what? Alteration or any change or any defect or any mutation in what? A single gene. Okay. If any change occur in a single gene, then that is called as Mendelian disorder or gene disorder, right? And these disorders are transmitted to offsprings on same lines as we have studied in the principles of inheritance and variations, right? They will be transmitted to the same line, that means to the next generation, right? The pattern of inheritance in such particular disorders will follow the Mendelian characters, that is why they are called as Mendelian disorders. Okay, the pattern of these particular genes will follow the Mendelian laws, that is why they are also called as what? Mendelian disorders. Clear? And they can be traced with the help of what? The pedigree analysis. They can be traced with the help of pedigree analysis. See, the pedigree analysis mainly is applicable for what? Mendelian disorders, right? And most common and most prevalent Mendelian disorders are what? The first one is hemophilia, second cystic fibrosis and sickle cell anemia and color blindness, then phenylketonuria and thalassemia. So these are the different disorders which are related to the defect in only one gene, that is Mendelian disorder. Okay, these are the different examples for what? Mendelian disorder or gene disorder. So it is important to mention here that such Mendelian disorder may be dominant or recessive. Okay. Here I have given the examples of different disorders which are related with what? Only one gene, or which are occurred due to defect in only one gene. And these particular disorders which are occurring due to defect in only one gene may be dominant or may be recessive. It means the mutation which has occurred in these particular genes, the mutation may be dominant mutation or it may be recessive mutation. See here, I will just give you the brief account of mutation. Okay, the mutations which may be dominant or which may be recessive. Do remember that when you take any of the genes or alleles, okay, say for example A and A, in normal condition it is called as wild type. Okay, in normal condition it is called as wild type. If any of this undergo mutation, okay, say for example A and A have undergone mutation to form A and A dash, the mutation occurred with this particular allele. When the mutation have occurred, if the character is altered, even in presence of the normal gene, then it is called dominant mutation. Okay, if the character is altered, then it is called dominant mutation. Even in presence of a normal gene, okay, if the mutated gene get expressed, even in presence of normal gene, if the mutated gene get expressed, then it is called as what? The dominant mutation. In case of dominant mutation, what will happen? The mutated gene will be dominant, but not the wild type. That type of mutations are called as dominant mutations. And it may also occur that if in case of B and B, okay, if one undergoes mutation to form B dash, then what will happen? It may be dominant. If it is dominant, then the character will be expressed, as I have told in the first example. But it may also be recessive. It means that in a presence of normal gene, the mutated gene will not express, or it will be suppressed. This type of mutation is called as the recessive mutation. Okay, in presence of normal or wild type of the allele, the mutated allele will get silent or will not be expressed. This type of mutation is called as what? Recessive mutation. Okay, recessive mutation. Clear? And to remember, most of the mutations that occur will be of what? Recessive type. Most of the mutations that occur will be of what? Recessive type, but few of them can be what? Dominant in nature. Okay, and by means of pedigree analysis one can easily understand whether the trait in the question is dominant or recessive, because in case of dominant trait, as we have already came to know the rules of pedigree analysis, you have seen in case of dominant trait or dominant mutation, skipping of generation is not seen. Okay, if skipping of generation is not there, then you can conclude that the type of mutation is dominant. Okay, if skipping of generation is said, then it is a recessive type of mutation, right? Okay, now let us go to these particular diseases or gene disorders one by one. Coming to the first one, that is hemophilia. Okay, hemophilia is an example for Mendelian disorder. It means this particular disorder is related to only one gene. See, and this particular gene which is responsible for the disorder hemophilia is what? Sex linked. It means the particular gene which is responsible for this particular disease is present on the X chromosome. It is present on X chromosome. Okay, how can I say that it is present on X chromosome? Because it is showing sex linked recessive nature. If it is given just sex linked, then it will be a confusion. Since they have given clearly here it is recessive, okay, you can say that it is X linked recessive. Okay, it is what? X linked recessive. Because if it is Y linked, in case of Y linked, there is no recessive and there is no dominant condition, because the Y chromosome, okay, because the Y chromosome occurs alone, there is no homologue for that. But X chromosome will have one homologue. In case of females, the X chromosome will have the homologue. That is why possibility of dominancy and recessivity is possible. Okay. Next, hemophilia is a sex-linked recessive disease that shows transmission from unaffected, that is carrier female, to some of the male progeny. Okay, some of the male progeny from carrier female. Okay, carrier means what? Unaffected. See here, X and X dash. Here X dash is what? The mutated form. Okay, and this mutated form is what? Recessive. Okay, this mutated form is recessive. Since it is recessive, in the individual, in the female individual, it cannot be expressed. Okay, since it is a recessive type of mutation, in presence of a normal gene or normal allele, it will not express itself. Okay, since it cannot express itself in the presence of normal allele, okay, it is a recessive type of mutation, and the individual will become what? Carrier for the particular disease or disorder. The individual will become what? Carrier for particular disease or disorder. And that carrier mother, okay, the carrier female or carrier mother can transmit this disease to the male progeny, because she will be giving what? X chromosome to the male individual, right? That is why 50% of chances will be there. Okay, 50% of chances will be there of what? Getting the infection in the male progenies produced by the female. Okay. Next point, in this disease, a single protein, very important point, a single protein which is a part of cascade of proteins. It means single protein from a cascade of protein. Here cascade means group. Okay. In this disease, what happens? Single protein from a group of proteins involved in clotting factor is affected. Okay. In this particular disease, what happens? The single protein that is a part of group of proteins, okay, which are involved in clotting of the blood will be affected. It means a gene that is responsible for clotting factors, production of clotting factors will be affected. Okay. Next, due to this infection or due to this defect, what will happen? In the infected or in the affected individual, okay, even a simple cut will result into non-stop bleeding. Okay, even a single cut, even a small cut on the skin will lead to continuous bleeding, because the clotting factors, since they are affected, clotting of the blood will not take place very immediately. Okay. Since the clotting of the blood do not take place, then it will lead to what? Continuous bleeding, right? And next point, heterozygous female who is called as a carrier, okay, for this particular disease may transmit the disease to the son. Okay, heterozygous female, that is X and X dash, she is heterozygous, which is called as carrier. Okay, she is called as what? Carrier. She may transmit the disease to their sons. Okay, she may transmit the disease to their sons, that too particularly half of the sons. It means if she has got 2 sons, there may be possibility of one son getting this one, because other son may be having the normal X chromosome which will be transmitted. The possibility will be there 50-50. Okay, 50-50 possibility will be there. Clear? Next one, possibility of female becoming hemophilic is extremely rare. Okay, why? Because, okay, I will write it very clearly here. See, possibility of getting the infection in the female individual, okay, possibility of the female being hemophilic, okay, is extremely rare. Why? Because if the female should get affect, okay, if the trait is occurring in the female, then, or if the female to get affected, she should have the homozygous condition. Okay, only in case of X dash and X dash condition she will get the particular disease. Is that clear? And if you want to have this condition in the female, then what should happen? The marriage between carrier female with, okay, affected male should take place. Yes or no? The marriage between carrier female and affected male, then only, okay, it is possible to get the homozygous condition in the females. That is why this type of marriages are rare. Okay, this type of marriages are possibly rare. That is why the possibility of getting infection or possibility of getting the disease in the female will be extremely rare. See, they have given, because mother of such female has to be at least carrier and the father should be hemophilic. Okay, here mother is carrier and father is hemophilic, because the X dash or mutated gene is present. Okay, mutated gene, if it is present, there is no normal gene to suppress it. Okay, because the other chromosome is what? Y here. Since Y is there, okay, there will be no gene to suppress the mutated one. Okay, if it is X here, then it could have suppressed, but in the males both the X chromosomes will not be present, only one X chromosome will be present and other will be Y. That is why there is no other homologue to suppress the same thing. Hence the male will become affected, right? And if the marriage happens between affected male and the carrier female, there is possibility of getting what? The infection or the affected condition in the females. Is that clear? Next one, the family pedigree of Queen Victoria. Very important point to be noted here. The family pedigree of Queen Victoria shows number of hemophilic descendants as she was a carrier. Okay, as she was carrier. Hence, this disease is also called as royal disease. Okay, royal disease, because this disease was found in the royal family. Okay, royal family, hence it is called as what? Royal disease. Okay, and since the individuals here show continuous bleeding or non-stop bleeding, this disease has got another name, that is bleeder's disease. Okay, bleeder's disease. It is also called as what? Bleeder's disease or the royal disease. Clear? And very important point to be noted in this one is, it is sex linked recessive. Okay, because in the pedigree analysis when they give, okay, you should remember that hemophilia is sex linked recessive one. Okay, in the pedigree analysis you may get this type of question. Okay, next one, coming to the next one, that is sickle cell anemia, the another Mendelian disease or disorder. Okay, Mendelian disorder or gene disorder, that is sickle cell anemia. And important point about sickle cell anemia is, see, it is an autosome linked recessive trait. Okay, autosome linked recessive trait. It means this particular disease is related with what? Autosomes. Okay, autosomes are nothing but the body chromosomes, not the sex chromosomes. Is that clear? The body chromosomes. It means the trait can be seen with equal frequency in both males and females. If it is sex linked, then the traits may vary in males and females, but here it is autosome linked, hence it can be seen in both males and females with equal frequency. And the next point is, the gene for this particular disease is located on autosome. Okay, autosome means the body chromosome, right? And since it is autosome linked recessive trait, it can be transmitted from parents to the offsprings when both the parents are, see, when both the parents are carrier for the gene. Okay, when both the parents are carrier for the gene, carrier means heterozygous. It is occurring in the individual only if both the parents are carriers. Say for example, if the father is having A and A dash, who is a carrier for this autosomal disorder, and if he marries, or if the mother is also a carrier A and A dash, then only possibility of getting A dash and A dash is possible, or the possibility will be what? Present. Okay, the possibility of getting A dash and A dash will be present. Is that clear? That is why the individuals which will get the sickle cell anemia, their parents will be, both of their parents will be carriers. If only one parent is carrier, then possibility of getting the disease is 0. Okay, if only one parent is carrier, then possibility of getting the infection is 0. Okay, that means one if it is carrier and another parent if it is normal. Okay, but when one is carrier and other is affected, then what? The offsprings which are produced, they will be showing what? The particular affected condition in a maximum number. Okay, because one is completely affected and other one is carrier, right? Possibility will be more. But if only one parent is carrier for the same and other is normal, then possibility will be 0. Okay, in getting the infection in the next offsprings or the next generation, the possibility will be 0. Clear? Next one, this disease is controlled by a single pair of allele, that is HBA and HBS. Out of the 3 possible genotypes, only homozygous individuals for HBS, it means homozygous means HBS and HBS, that shows the diseased phenotype. Okay, but heterozygous condition, that is HBA and HBS individuals, appear apparently unaffected, but they will become the carrier of the disease. It means only when the condition is HBS and HBS, okay, in homozygous condition, the infection can be seen. Okay, and in case of heterozygous condition, the individuals will remain unaffected and they will be what? Carrier in nature, right? And this defect is caused by, what is the main cause for this defect? See here, the defect is caused by substitution of glutamic acid by valine. Okay, substitution of what? Glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin molecule. Okay, hemoglobin molecule will be made up of alpha chain and beta chain. Okay, but in the beta globin chain, particularly on the 6th position, okay, if glutamic acid is replaced by valine, then this disease will be occurring. Okay, in a normal condition, the nature of RBC will be, okay, biconcave disc shaped. Okay, biconcave disc shape, like this the RBC will be biconcave, okay, and disc shape. This is the side wave and this is the top wave. On the top wave they will be disc shape, on the side wave they will be biconcave in shape, right. So in case of the normal condition, the RBC will have a biconcave disc shape, but in these individuals who have got sickle cell anemia or in the affected condition, the biconcave shape of the RBC will be lost and they will lead to what, or they will have sickle shaped RBC, okay, sickle shaped RBC, and due to sickle shaped RBC they will show what, less supply of oxygen to all their body parts, okay, they will be anemic in nature, right. Next one, substitution of amino acid in globin protein results due to single base substitution at 6th codon of the beta globin chain. Is that clear? On the 6th particular codon, what will be there? Okay, GAG will be there and it will be replaced by what? The GUG. Okay. See here, the substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin chain from GAG to GUG. Okay. The glutamic acid will be replaced by valine. See here, the change occurs in only one position, okay, at only one point. That is why it can be called as point mutation. Okay, and the change is occurring in only one gene, hence this is an example for gene mutation. Is that clear? And next one, the mutant hemoglobin molecule undergoes polymerization, okay, polymerization under low oxygen tension, and due to polymerization of the hemoglobin under low oxygen tension will cause what? Change in the shape of the RBC from biconcave disc to elongated or sickle shaped in nature, okay, from normal biconcave disc shape to, okay, the shape will changes into what? Elongated sickle like structure. So this particular disease is termed as what? Sickle shape or sickle cell anemia. What is sickle cell anemia? It is autosomal recessive disorder which is resulted by means of what? Substitution of glutamic acid by valine. Substitution of glutamic acid by valine, and substitution of glutamic acid by valine will take place in which particular place? At the 6th position of beta globin chain. Okay, and why the glutamic acid is replaced by valine? By the mutation in only one base. Okay, in what? The 6th codon of beta globin chain. Okay, due to which what will happen? The hemoglobin molecule undergo polymerization under low oxygen. Okay, very important point. The polymerization of hemoglobin will undergo what? Or polymerization will undergo in what? Low oxygen content or low oxygen tension, which will cause the change in shape of the RBC from biconcave disc shaped to what? Sickle shaped. So that is about what? Sickle cell anemia. Next one, coming to the next one, that is phenylketonuria. Okay, the next disease is phenylketonuria. And what is phenylketonuria? See here, it is inborn error of metabolism. Very important point. It is inborn error of metabolism which is inherited as autosomal recessive trait. Okay, it is also an example for what? Autosomal recessive trait. What is autosomal recessive trait? It means the gene for this particular disorder is located on the autosome, not the sex chromosome, the autosome. Is that clear? Next, what happens to the affected individuals? They lack an enzyme that converts amino acid phenylalanine into tyrosine. Okay, phenylalanine will be converted into tyrosine in the normal individual. Okay, the amino acid called as phenylalanine will be converted into what? Tyrosine in the normal individuals. But in these individuals having the phenylketonuria, what happens? The enzyme which is responsible for conversion of the phenylalanine into tyrosine will be absent. As a result, the phenylalanine is accumulated and converted into what? Phenylpyruvic acid and other derivatives. Okay, due to accumulation of phenylalanine, okay, what will happen? The accumulation of phenylalanine in the form of phenylpyruvic acid and other derivatives, what will happen? It will lead to, okay, the phenylketonuria. Okay, it will lead to phenylketonuria, and it results in what? Okay, the mental retardation. Accumulation of these derivatives results in mental retardation of the individual. These also secreted through, that means these phenylpyruvic acid and its derivatives are also excreted through what? The urine, okay, because of their pure absorption by the kidney. By means of this urine test, okay, by means of urine test, this particular disease can be identified. Okay, in the urine, if the derivatives of phenylpyruvic acid or the phenylalanine, if they are seen, then it can say that the particular condition is phenylketonuria. And here important point to be noted is what? It is an autosomal recessive trait, okay, autosomal recessive trait, and what happens here? An enzyme responsible for conversion of the phenylalanine into tyrosine will be absent. Okay, it will not be produced. Due to this one, what will happen? Accumulation of phenylpyruvic acid and other derivatives will be taking place. Right? Next one, thalassemia. Okay, it is another disorder related with what? The blood, again the blood, right? And see here, it is an autosome linked recessive disorder. Okay, again it is autosome linked recessive. As I have already told you in the beginning, most of the gene disorders will be showing a recessive condition. Okay, like the same, it is the third one which we are studying, okay, that is showing autosomal recessive nature, right? And this is an autosomal linked recessive blood disease, okay, transmitted from parents to the offsprings when both the parents are what? Unaffected, but they will be carrier. Okay. In case of autosomal recessive disorders, do remember, autosomal recessive disorders, if you want to have an affected individuals, then both the parents should be what? Carrier for the same, or else one should be carrier and other should be affected. So in that case, what will happen? The affected individuals can be obtained. See, this defect is due to either the mutation or deletion, which ultimately results in a reduced rate of synthesis of one of the globin chains, that is alpha or beta. Okay. In the globin chains, there are two types. I have already told you in the hemophilia condition. While discussing about the hemophilia, I have told you what? The particular hemoglobin molecule will be made up of two globin chains, that is alpha globin chain and beta globin chain. And the thalassemia may be resulted by means of what? The mutation or deletion in either one of these particular globin chains, either beta globin chain or alpha globin chain. If any of them are having the defect, then this particular thalassemia condition can be seen. Next one, this causes, it means the thalassemia causes what? Abnormal hemoglobin molecules, okay, which results into anemia. Anemia is nothing but what? Lack of proper hemoglobin content, okay, lack of proper hemoglobin content. And the thalassemia can be classified according to which chain of the hemoglobin is affected. If it is affected with alpha chain, then it is called as alpha thalassemia. If it is affected with the beta chain, then it is called beta thalassemia. Okay. If alpha chain is affected or showing the mutation, it is termed as alpha thalassemia, and if the beta chain is mutated, then it is said to be beta thalassemia. Right. Next one, see. So in case of alpha thalassemia, it is controlled by two closely linked genes, that is HbA1 and HbA2 on the chromosome number 16. So very important point for your CET and NEET examination, because it is the disease which is added in the new textbook of second PUC. Second PUC in the NCERT, in the new textbook, this particular disease is added. Okay, the thalassemia is added this year or the previous year, right? So here you should remember the thalassemia is resulted by means of the change in alpha chain or beta chain. In case of alpha thalassemia, it means where the alpha chain is added, alpha chain is showing the defect. Okay, what happens in this one? The alpha thalassemia, it is controlled by what? Two closely linked genes. They are HbA1 and HbA2. Okay, and these genes will be present on 16th chromosome. Okay, 16th chromosome of each parent, and it is observed due to what? The mutation or deletion of one or more of these four genes. Okay, HbA1 or HbA2. Next one. The more genes affected, then less alpha globin molecules are produced, while in case of beta thalassemia, it is controlled by single gene. See, alpha thalassemia is controlled by two closely linked genes, that is HbA1 and HbA2, but beta thalassemia is controlled by only one gene, that is HbB. And see here, the alpha thalassemia, that is resulted by means of change in the HbA1 or HbA2. It is present on 16th chromosome, but in beta thalassemia, the particular gene is present, that is HbB is present on chromosomal number 11. On 11th chromosome it is present, and due to the mutation of one or both genes, the beta thalassemia will be resulted. Okay, next one. Thalassemia differs from sickle cell anemia in that former is, okay, in the condition where the former is quantitative problem of synthesizing too few globin molecules, while the later is qualitative problem of synthesizing an incorrectly functioning globin. Okay, here the difference between what? Sickle cell anemia and the beta thalassemia or the thalassemia is such that the qualitative and quantitative difference will be seen. Okay, qualitative and quantitative difference will be seen. That means number of hemoglobins will be produced in less quantity, and in first one, that is in sickle cell anemia, the quality of the hemoglobin which is produced will be altered. In sickle cell anemia, the quality will be altered because the shape of the RBC will be lost there from biconcave disc shape to what? Sickle shaped, right? Next one. The next one is color blindness. Okay, the next one is what? Color blindness. So it is a common disorder, okay, the common genetic disorder or gene disorder which is seen, okay, and this disorder is sex linked recessive. Okay, it is what? Sex linked recessive disorder. The color blindness is sex linked recessive disorder due to the defect in either red or green cone, okay, red or green cone of the eye, which results in failure to discriminate or failure to differentiate between red and green coloration. Hence the color blindness type is called as red and green color blindness. Okay, hence it is called as what? Red and green color blindness. Is that clear? And this defect is due to what? Mutation in certain genes present on the X chromosomes. Okay, due to the mutation in certain genes present on X chromosome. And very importantly, see here, it occurs about, okay, it occurs in about 8% of males and only about 0.4% of the females. Why? Because it is X linked. Okay, it is X linked recessive disorder, and the X chromosome is only one in the males because another one will be what? Y. Due to that, in case of males, what is happen? Okay, the level of expression of this will be more, or the possibility of getting the infection will be more. But in case of females, what will be there? X and X condition will be there. Okay, X and X condition. If this is the condition in the females, that is if the affected gene is present on one X chromosome, the normal gene will be there which will suppress it one, okay, which will suppress this one. Clear? By means of that, the females will become carrier and the males will get the infection. Okay, if you want to see the females getting infected by this one or getting affected by this one, then what should happen? The marriage between carrier female with a affected male should take place. Then only the possibility of getting infected females can be seen. Clear? That is why the possibility of having the infection in the males is 8%, but in the females it is around just 0.4%. That is extremely rare. It is because the gene that lead to red-green color blindness are on the X chromosome. Males have only one X chromosome. The same thing whatever I have explained is given in points. See here, why it is more in males? Because males have only one X chromosomes and females have two. And son of a woman, okay, son of a woman who is a carrier for this particular gene, okay, will be having the chances of what? 50% of getting the infection. Okay, if the mother is carrier, see here, if the mother is carrier and marries to a father, okay, then what will happen? The male individuals produced by this will be X and X and X dash and X. Yes or no? That is why out of 100% males produced by this one, 50% will be having the chances of getting the infection. Okay, out of 100% possibility, 50% will be having the chances of what? Getting the infection. Right. The next one, mother is not herself color blind because the gene is recessive and she will become what? Carrier for the same. And daughter will not normally be color blind unless her mother is a carrier and her father is color blind or affected, as I have shown in this one. Clear? Okay. So, this is about what? The color blindness. Let us go to the next one, that is chromosomal disorders. Okay. The next one is chromosomal disorders. What exactly happens in chromosomal disorders? These are the disorders caused by what? Absence or excess or abnormal arrangement of one or more chromosomes. Okay, these are the disorders which are related to the complete chromosome. Okay, and if the change is seen, okay, what may be the change? Absence or excess or abnormal arrangement or deletion of a part. Deletion of a part of the chromosome. Okay. Okay, deletion of a part of a chromosome. So, then what will happen? The chromosomal disorders can be seen. Why the chromosomal disorders occur? Because they occur due to failure of segregation of the chromatids during cell division. When the chromatids are getting separated due to improper separation or due to improper segregation, what will happen? The loss or gain of a chromosome or a part of it will be seen. Okay, that will lead to what? Occurrence of aneuploidy. Okay, say for example, in the Down's syndrome, okay, in the Down's syndrome where the individual gain an extra copy of chromosome in the 21st chromosomal set. Okay, normally the 21st chromosome will be having only 2 alleles or 2 homologous chromosomes. Along with these chromosomes on the 21st chromosomal set, okay, along with the normal chromosomal set, what will happen? Another chromosome will be occurring. So, this is called as 21st trisomy. Okay, it is called as 21st trisomy, or it is also called as autosomal trisomy. Okay, it is also called as autosomal trisomy. Why? Because what is a trisomy condition? It is a condition in which 3 chromosomes are occurring in a particular region, right? Instead of normal 2 chromosomes or a pair of chromosomes, what will happen? Another chromosome will come and sit there along with the pair, which will lead to occurrence of 3 chromosomes. That is called trisomy. And since it is occurring with the 21st chromosome, which is an autosome, hence it is called as autosomal trisomy, right? And similarly, Turner's syndrome results into loss of an X chromosome in the human females, which is called as what? Okay, the allosomal monosomy. Okay, allosomal monosomy. Why? Because here in case of the Turner syndrome, what will happen? Along with the 44 autosomes, what will happen? Only one X chromosome will be there. There is no other chromosome. Okay, there is no other chromosome. That is why allosomal means sex chromosomal monosomy means only one sex chromosome will be present because another X chromosome is lost, right, which is called as allosomal monosomy. Next one, failure of cytokinesis after telophase stage of cell division results in increase in the whole set of chromosomes in an organism, and this is known as what? Polyploidy. Okay, polyploidy, which is commonly found in what? The plants. What do you mean by polyploidy? Very simple. If only one chromosome is increasing or decreasing in nature, then they may be called as what? Autosomal or allosomal monosomy or trisomy. But if a complete set of the chromosome is increased, then it will lead to increase or decrease in the ploidy, that means complete set of the chromosome. So what do you call it? N as it is haploid. In haploid, one complete set of chromosome will be there. If two sets of chromosomes are there, then it is called diploid. Okay, and if another set of chromosome occur along with this, it will be leading to formation of 3N, which is called as triploid. Okay, like this, if many sets of chromosomes, okay, complete sets of chromosomes occur together, okay, it is called as polyploidy. Poly means many, ploidy means set of chromosomes, the complete set of chromosomes. It is called as what? Polyploidy. Clear? It is called polyploidy, and the polyploidy condition is commonly seen in case of what? Plants, not the animals. Why the polyploidy occurs? Because of failure of cytokinesis. What do you mean by cytokinesis? Division of the cytoplasm. Okay, division of the cytoplasm, if does not happen properly, then what will happen? Two sets of chromosomes will remain as it is in the cells, in the same cell, then it will lead to polyploidy condition, right? Next, see here, total number of chromosomes in a normal human cell is 46. Okay, that is 23 pair. Out of these, 22 pairs are autosomes. That means they are related with what? The control of body characters, and other two are what? The sex chromosomes, which are also called as allosomes. Okay, which are also called as allosomes, right? Though rarely, either an addition of a copy of chromosome may be included in an individual, or an individual may lack any of one pair of the chromosome. Okay, and such conditions we are going to see further here. So let us go through them one by one. The first one is Down's syndrome. Okay, Down syndrome. It is called as autosomal trisomy. Why it is called autosomal trisomy? Because here what happens? It is caused due to the presence of an additional copy of chromosome in the chromosomal number 21. That is why it is called as trisomy of 21st chromosome, or since the 21st chromosome is autosome, we can call it as autosomal trisomy. In the individuals having the Down's syndrome, or individuals who have got the Down syndrome, they will have one extra chromosome along with the pair in the 21st chromosomal set. The 21st chromosomal set will normally have two chromosomes. Along with that, one extra chromosome will be present. So this will be called as what? Trisomy of 21st chromosome, which is called as autosomal trisomy. Okay, this was discovered by Langdon Down in the year 1866. Hence, the disease is given with the same name called as Down's syndrome. Okay, hence it is called as Down's syndrome. Clear? Here affected individual is short statured, okay, with small and round head, with furrowed tongue and partially open mouth. These are the characters of what? The Down's syndrome. The affected individuals will be short statured. He will be not having the proper normal height. Okay, the height will be lessened or short, and small round face will be there, and furrowed tongue and partially open mouth. And the most important point here is the palm is broad with characteristic palm crease. Okay, when you observe the palm of the individual, okay, like this, a characteristic palm crease will be seen. This is what? The palm crease. Palm crease will be seen. A characteristic palm crease will be seen here. Right? Next one. Physical as well as psychomotor and mental development is retarded. Very important point here. In the Down syndrome, okay, the individuals may be mentally retarded. Okay, individuals may be mentally retarded. In severe conditions, the individuals will be having physical, psychomotor as well as mental retardedness, okay, or defect in physical, psychomotor and mental development. Right? This is about what? Down's syndrome, which is an autosomal, okay, trisomy condition or autosomal chromosomal disorder. Okay. Next one, the Klinefelter syndrome. See here, what is Klinefelter syndrome? It is allosomal disorder, okay, allosomal disorder where in which allosomal trisomy can be seen. Okay, allosomal trisomy can be seen. That means in the sex chromosomes, okay, in the sex chromosomes, set of 3 chromosomes are seen here. Okay, set of 3 chromosomes. Actually, in the males, along with 44 chromosomes, 44 autosomes, they will have one X and one Y chromosome. But in the Klinefelters, okay, in the Klinefelters, what will happen? Allosomal trisomy will be seen, wherein which the chromosomal content will be 44 XXY. Here one X chromosome will be added extra. That will result in what? Allosomal trisomy. Okay, additional copy of X chromosome results into what? A karyotype of 47. It means total number of chromosomes will become what? 47. So that condition is called Klinefelter syndrome. And such individuals have overall masculine development. That means the body of the individual will be male like, okay, masculine development, but they will show the feminine characters such as development of the breast. Okay, the condition is called as gynecomastia. Okay, gynecomastia. It means development of the breast in the male individuals. Okay, due to presence of an extra X chromosome. Okay, in the Klinefelter syndrome is called as gynecomastia. Right? And such individuals will be sterile in nature. Okay, such individuals will be sterile in nature. It means they cannot produce the normal sperms, or the spermatogenesis will not be seen. The spermatogenesis will be absent. Right? That is what? The Klinefelter syndrome. And do remember why this Klinefelter syndrome occurs? Due to occurrence of an extra chromosome. Why the extra X chromosome occurs? During the gamete formation, say for example 44 X and X will be there, and in the male 44 X and Y will be there. During the gamete formation, what happens? 44 X should be divided into 22 X and 22 X. Okay, but in case of this condition, the proper disjunction will not take place, that lead to 44x x into what? 22x and x here, but another one will be 22x, 22 without any chromosome, right? So this is how, or else it may happen in the males also, that is 22x and y will come in one particular sperm and other sperm will not have any chromosome, okay, that is sex chromosome. These are the conditions, okay. Due to improper or non-proper disjunctioning, Okay, this particular condition will be seen. So next and the last one, that is Turner syndrome. It is allosomal means it is also sex linked. Okay, allosomal monosomy. Okay, it is allosomal monosomy, where what happens? The sex chromosome will have only one, that is only one chromosome. Okay, such disorder is caused due to absence of one X chromosome, which will lead to 45 condition. Okay, here the individual will have 44 and X. Okay, 44 autosomes will be normal, but sex chromosome will be only one here. There is no other sex chromosome. Okay, the other sex chromosome will be absent. Is that clear? So this condition where other sex chromosome is absent, okay, so this will lead to what? Only presence of 44 autosomes with one X chromosome, which will lead to what? 45. So this condition is called as Turner's syndrome, right? And such females, okay, in such individuals the body will be females or feminine. The body will be feminine, but those feminine or females will be what? Sterile in nature. They will be sterile in nature. Okay, what is the reason behind their sterility? Okay, the ovaries are rudimentary or underdeveloped. Okay, the ovaries are underdeveloped, and even it includes what? The lack of other secondary sexual characters such as the breast development. Okay, breast development and the menstrual cycle. All of these, which are the secondary sexual characters, will be absent in these particular individuals. Okay, here this is also resulted by means of what? Improper non-disjunctioning of the chromosomes. Okay, this also occur due to what? Improper, okay, disjunctioning of the chromosomes. Okay, by means of this we shall finish today's video, and this is the end of what? The theoretical part of the chapter Principles of Inheritance and Variations. Okay, in the next video we shall start with the discussion of the questionnaire. Okay, let us see in the next video. Thank you.